Hello and welcome to our second video of our Carl Chat series. In the first video, our Chief Scientific Officer, Dr. Carl Ascoli, and our Product Manager, Todd Giardello, discussed COVID-19 and the effects it had on the biotech industry. Today, they'll be discussing recombinant antibodies, what they are, how they're produced, and the advantages they provide to the IVD and therapeutic markets. We hope you enjoy the discussion. Let's begin.
Thank you everyone again for joining us with another Carl Chats. With me is Dr. Carl Ascoli, and today we're going to be talking about recombinant antibodies. So Carl, as a kickstart, what are recombinant antibodies?
Good morning, Todd. Well, very simply, recombinant antibodies are monoclonal antibodies with a defined genetic sequence. So not only is the nucleic acid sequence of the antibody known, the amino acid sequence of the antibody is also known, and it can potentially be produced without generating an immune response in a host animal, potentially.
That's interesting. So then how are recombinant antibodies produced?
Well, there's no one way to produce a recombinant antibody. In fact, there's many ways. One of the most common ways is to clone the nucleic acid from, for instance, the B cells of a hyperimmunized animal, or from a hybridoma cell line that's been produced and subcloned. And in both of those instances, actually, the recombinant antibody is originating from an animal source. Typically, what happens is when you make a recombinant antibody is after you clone it, you put it into an expression library for selection and subcloning. In some instances, you can make a recombinant antibody that is animal-free. For instance, you can directly synthesize a nucleic acid and then express the protein that makes the recombinant antibody.
So then how does the manufacturing process of recombinant antibodies differ from something that we would expect to be a more traditional monoclonal antibody?
Well, in general terms, making a recombinant antibody is more difficult because there are more steps involved. And therefore, the cost of generating a recombinant antibody is higher compared to a conventional hybridoma-based monoclonal or a polyclonal antibody. But if the antibody that you're producing is needed in large quantities, if there's lots of demand for that material, and if in fact you're going to use it over a long period of time and generate multiple lots, then the cost and difficulties in producing the recombinant are outweighed by eliminating the costs of remanufacturing and the difficulties that go along with remanufacturing.
So how do recombinant antibodies compare to the traditional Maureen-derived monoclonal antibodies?
Sure. Well, conventional hybridoma-based monoclonals are produced by well-defined steps. You hyperimmunize an animal, you fuse the splenocytes with the hybridoma, with a myeloma cell line to make the hybridoma, and then you go through subsequent rounds of subcloning and screening until you get a truly monoclonal cell line. As for many cells, hybridoma cells are subject to cell death, chromosomal rearrangement, and clonal drift. So even though you make the hybridoma cell line, things could change over time. And conventional hybridoma-based monoclonals don't have a defined genetic sequence. And that is an issue in many areas as it relates to therapeutics and reproducibility.
So how does that differ from recombinant monoclonal antibodies?
Well, a recombinant antibody can be produced again with a known genetic sequence. That's one of the big differences. And therefore it can be produced and potentially reproduced in an animal-free environment. With the known genetic information, you can use conventional molecular biology techniques to produce the proteins with the same sensitivity, specificity, and reproducibility every time you produce the antibody.
So how about what are some of the key benefits to using recombinant antibodies?
Sure. Well, again, in my opinion, recombinant antibodies are very well suited for long-term use where multiple lots are going to be required to minimize any performance differences over a long period of time. I think recombinant antibodies are best suited for the diagnostic and the therapeutic uses. In fact, most therapeutic antibodies today, if not all, are in fact recombinant antibodies. And one of the most significant benefits of recombinant antibodies is that the sequence itself can be engineered. So you can modulate the properties, you can optimize the properties of the antibody with regard to performance and stability, depending on what immunoassay you're configuring.
So you had mentioned that there are some advantages, particularly to the diagnostic and therapeutic industries. Can you expand on that a little bit more?
Well, sure. When an antibody is used for diagnostic purposes, typically it's needed in a large quantity, and it may have requirements of being needed over a long period of time, and you may need multiple lots. And in all of these conditions, you really have to have an antibody that is invariant from one lot to another and from one year to the next. So recombinant antibodies are really well suited here. In therapeutics, the argument is even stronger for using recombinants because of the fact that you have a known genetic sequence. And regulatory agencies, again, consider recombinant antibodies as drugs, and you really have to define the basic composition of a drug. So that's a very strong point for using recombinants for therapeutics. And also, as I mentioned before, the ability to engineer the sequence gives you a strong advantage. You can modulate the sensitivity, the specificity, and even the stability of a drug. For instance, you can deploy a therapeutic that might be cold chain independent in a particular use.
So we've been talking a lot about the benefits of recombinant antibodies, but are there any limitations to their production and performance?
Yeah, there are technical limitations, and I did mention earlier that initially there could be some cost considerations as well. For instance, technically, if you are producing a recombinant antibody that has a heavy and light chain like a conventional antibody, there may be large libraries that need to be constructed in order to obtain an antibody with the properties that you want. On a cost basis, if you're doing research, for instance, some antibody targets and antibodies themselves may be better suited to first producing a conventional polyclonal antibody, which is very cost effective to produce. And once you in fact determine that there's biological significance to the target when you're doing research, and that antibody is going to be used by a lot of people, then there may be the justification to make a recombinant antibody. But I happen to be one of many people that think that polyclonal antibodies are very valuable tools as well.
Are recombinant antibodies better suited for any immunoassay applications more so than, say, polyclonal or monoclonal antibodies?
My answer to that would be generally no. However, if the recombinant antibody is a single domain antibody where there's only one binding event, then any immunoassay that requires bivalent antibodies for binding may not work well compared to an antibody configured with a heavy and light chain. Again, I'm of the opinion that recombinant antibodies work best for diagnostics and therapeutics, and I think they show less of a bias one way or the other, depending on the immunoassay that they're going to be used in.
So let's conclude with some market trends. The recombinant antibody market is expected to increase by about 7% for the next few years, while the total antibody market is expected to increase about 11% during that same time. Do you expect the differences in these trends to continue, or will the benefits that we've talked about today of recombinant antibodies sway immunoassay manufacturers in their direction?
Sure. I guess my answer would be yes. I think recombinant antibodies will continue to gain market share. There are trends, specifically in Europe right now, to embrace an animal-free approach to antibody production, and that may actually accelerate the rate of growth of recombinant antibodies in the marketplace. But remember, many of the strategies for producing recombinant antibodies actually start with libraries created from hyperimmunized animals. So the perceived gain from an ethical perspective may not be as advertised because ultimately, a lot of these recombinants do derive from animals.
All right, so there's a lot of interesting information in there. I'd like to thank Dr. Carl Ascoli again for joining us today, and we hope to see you all again later with another Carl's Chat. Thank you, Carl.
Thank you, Todd. It was enjoyable. Talk to you next time.
All right, thank you.